Pleasure to sorry. It's my great pleasure to welcome Steve Michell to come to talk to us about today for vaccines for hospital-acquired infections. So Steve is a senior lecturer in molecular microbiology and biosciences within the university. And I know that you know Bea Knight very well, who's the co-lead for the PPI group with me. And I know she's very disappointed that she isn't able to be here today. But it's my great pleasure to welcome you to come and talk to us about vaccines for hospital-acquired infections. So thank you very much, and I'll pass it over to you.
Okay, thanks, Kim. Can everyone hear okay? Thumbs up or yeah? Okay, great. So I'll give a bit of background about me. I'm not that new to academia, really. I've been here 14 years, but I came down here when I was 37 after having been at the Defence Science Technology Laboratories at Porton Down looking at vaccines for bioterrorism agents. And before that, I worked for the Animal Plant Health Agency at Weybridge looking at vaccines for tuberculosis, specifically bovine TB. And before that, I'd done my PhD at St Mary's Hospital, London on human TB. And before that, I did my degree in chemistry and biochemistry at Imperial College London. So I'm now here in Exeter working on a variety of things, but I've had a lot of involvement with the hospital, especially some great clinical microbiologists there. And the bacteria of interest that I look at is something called Clostridium difficile. It's now called Clostroides difficile, but I'll come on to that later in the talk. So what I'm going to do, first of all, is just give you a bit of an overview about vaccines, what they are. I mean, vaccines are massively topical at the moment over the last 2.5 years for something we're all aware of. But if we go back all the way back to 1796, I do find it quite odd that apparently the French are very hesitant in having this SARS-CoV-2 vaccine at the moment. And given that France has such a big history with vaccinology, it's quite an odd stance that they've taken. But actually, it was in Gloucestershire that the first vaccine, or the idea of vaccination, occurred. And this was by Edward Jenner, who realized that milkmaids were protected against getting smallpox when they had cowpox on their hands. And what he did was lance some of these boils on the hands of milkmaids and took that material and ministered it to a little boy and showed that he was protected against smallpox. And that is really the history of the start of vaccinology. Things moved on then, and I don't know if you can see my mouse on the slide. So, I mean, for me, this is probably one of the most important developments in vaccinology. And this was in the early 1900s with two French vets, Calmet and Gorin, who developed the BCG vaccine for TB. TB is something that I personally think doesn't get half enough coverage. World Health Organization estimates that there are over 2 million deaths annually of TB. And that's when we're not really looking at it or recording it to the levels that we are for SARS-CoV-2 at the moment. And BCG was a live vaccine. And essentially what these vets did was take bovine tuberculosis, actually. And in those days, they didn't have little Petri dishes that we have in the lab now. And they cultured it on on potato slices, soaked in ox bile. And then they just looked at the morphology of this bacteria or the bacterial colonies after a while and noticed that they changed morphology. It's a big leap, but what they did was took those bacteria, immunized rabbits with this, and then challenged them with TB. And lo and behold, the rabbits were protected, and that was the vaccine trial for BCG. The vaccine that's probably been given to more people on this planet than any other vaccine. It's probably going to get overtaken by COVID vaccines very soon, but you know, probably all of us here have had a BCG vaccine. I know that I certainly had a BCG vaccine when I was at school. So vaccines have been around for ages. That's all the point I want to make here. And just to allay some fears about vaccines, so if we look at this table here, or this diagram, you have anti-malarials, antibiotics, and these lines indicate when we first saw resistance to these agents following their introduction. So the introduction starts on the left-hand side, and then the right-hand side with the cross shows where we have failure or resistance to these products. So as you can see with antibiotics and antimalarials, we develop resistance fairly quickly. And it's not really surprising because antibiotics actually target essential processes of bacteria. So if it's essential and you're trying to kill it, it's going to develop resistance to it. And there's a big thinking that we should be looking at alternative ways of tackling bacterial infections, maybe understanding how they cause disease more and tackling that, something that's not essential for them to live. But if we look at vaccines, you know, we don't see resistance per se to vaccines. There are some failures of some vaccines, and that's just because the causative agent of the disease may change, and that the stimulating vaccine then might not confer protection. And we're all aware of this at the moment from media, where they say, are we going to be protected against the Omicron variant at the moment? But as a rule of thumb, vaccines are definitely the way to tackle infectious diseases. And this is just a slide that I've picked from the literature looking at a risk-benefit analysis for vaccines. So if you look at measles infection or vaccination with the measles vaccine and look at the possible outcomes. So if you have measles, the disease, you know, you have this percentage chance of getting all of these different symptoms. But if you've been vaccinated, you know, so it's up to you which one you want to do. Do you rather have the disease or the vaccination? And for many of the vaccines, it's clear that it's very sensible to take the vaccine. What I'm going to do now is just, hopefully this will work, is play a video just giving you an overview of what a vaccine is, because I think that was one of the questions. But I'm then going to elaborate on this, but it'll just give you some basic knowledge. It's only about a minute or two. Hopefully this will play, and hopefully you can hear the sound.
To understand how vaccines
Can
work,
you hear this?
it helps to look first at how the immune system works, because vaccines harness the natural activity of your immune system. There are about 100 trillion bacteria and viruses on your body. Not all of them cause disease, but some are able to get inside our bodies to multiply, and this can make us ill. There are barriers to stop this happening, but suppose some disease-causing bacteria do get through. Your immune system is quick to recognize them as invaders. This is because the proteins or sugars on the bacteria's surface have different shapes to any of the ones in the human body. They trigger a complex chain of events involving many different types of white blood cells working together. One type of white blood cell is able to make antibodies to fight the invaders. Antibodies can stick to the proteins or sugars on the bacteria's surface, and this kills the bacteria or disables them. However, not all antibodies will work against these bacteria. They have to be exactly the right shape, a bit like a key fitting a lock. Our bodies have a library of billions of white blood cells, each of which can make just one shape of antibody. Only a few of these antibodies will match the invading bacteria. Producing antibodies of the right shape can take several days. By this time, there could be billions of disease-causing bacteria in your body. Once the right cells are activated, they quickly divide and turn into a production line, making masses of antibodies that stick to the bacteria. Eventually, your body gets rid of all the bacteria and you recover. Antibodies remain in the blood, and some white blood cells may also become memory cells. If those specific bacteria invade the body again, the immune system will respond so quickly that you won't get ill. Vaccines work in the same way. They contain weakened or dead bacteria or viruses, or even just a few proteins or sugars from the surface. This is enough to convince the immune system that a real invader has got in. The same process takes place as when real bacteria or viruses invade our bodies, except you don't get ill. Afterwards, if your body ever meets the real thing, your immune system will remember it and get rid of it before you even know it's there.
So in a nutshell, that's what vaccines are. So what I'm going to do now is discuss the different types of vaccines in a bit more detail. So live vaccines, living but cannot cause disease. And the classic example of this is BCG, Bacillus subcalmatin for TB. It's an actual live vaccine. It's a live attenuated vaccine. So you have killed vaccines, and these are kills by heating or exposure to chemicals. And there are several bacterial diseases. Typhoid is one of them. There are new typhoid vaccines out there, but historically, heat-killed Salmonella typhi was used as a vaccine for typhoid. And you can kill these either by heating or exposure to chemicals. And one of the classic ways of inactivating a bacteria or a microorganism is by treating it with formaldehyde. But the problem with formaldehyde or formalin is that it may alter the structure or structural integrity of the bug, so it might not be as efficacious as a vaccine. Similarly with heating, though, that's a problem. You know, if you heat it, you can destroy the structure. It's all about maintaining the structure that the immune system needs to recognize. So this is why live attenuated vaccines are very good. This is what's hit a lot of the press at the moment, is these subunit vaccines or fragments of the microorganism, for example, proteins or polysaccharides. A great use for subunit vaccines, if we think about how microorganisms cause disease, if you think about tetanus, we've all had the tetanus vaccine. And essentially that is where you take the tetanus toxin, and that is then inactivated through formaldehyde inactivation, and then administered as a vaccine. And you mount an immune response, and you're protected against a tetanus. So it's very easy to use that approach, of this subunit approach, if you know that there's a specific agent that causes the disease. Think about cholera, it's the cholera toxin that causes disease. So to develop a vaccine against cholera, just take the toxin, inactivate it, and administer that as the vaccine. So that's why it's really important to understand how bugs cause disease, because then, you know, if you know that there's a specific etiological agent of disease, i.e. a toxin, you know, a toxin-based approach would seem sensible. But I'm going to talk about C. diff, where all of that theory breaks down with that microorganism. Over the last 10 years, 10 to 20 years now probably, there are these things being called DNA or RNA vaccines, and these make proteins after injection. I'm going to talk about this in a bit more detail. Before I go on to the hospital-acquired infections, I'm just going to talk about the SARS-CoV-2 or COVID vaccines, because they start to impinge on these new technologies at the bottom of this slide. So I'll start off with oh yes, so I've just had a slide here about the different types of vaccines. So if you have diphtheria, tetanus, this was coming back to the point I was making about if you know how a bug causes disease, through production of toxin, it's really quite easy to make a subunit vaccine. We're talking about the DNA RNA vaccine. So this is the Pfizer BioNTech vaccine, where you directly administer the messenger RNA. So all that is really is the genetic code that encodes for part of the virus. And in the case of COVID, it's the spike protein. So what they've done is they've taken the genetic message for the spike protein for COVID, wrapped it up in a little micelle, in a little ball of lipid, and then that gets injected into you. Those lipids then interact with your host cells. The DNA or the RNA enters the cell, and the protein's made, and then antibodies in the body recognize that made protein. So it's quite simple. A lot of the adverse effects that are seen with the Pfizer BioNTech vaccine, this messenger RNA vaccine, are actually through reaction to the lipids, not the mRNA. The other vaccine for COVID that everyone's probably heard of is the AstraZeneca vaccine. And this is where they use an adenovirus. So this is a weakened or non-pathogenic virus. So if we imagine this green virus here is SARS-CoV-2, and what they've done, they've taken out the gene for the spike protein, and then they've introduced that into the adenovirus, and then we get immunized with that weakened adenovirus. And what happens is then the gene, we use the host to make the spike protein, and then we make antibodies against it. So that's the basis of the two real vaccines for COVID. I just wanted to, this is the last slide I'm going to talk about COVID vaccines actually, and it's data that was given to me by a friend, Dr. Ashley Otter at Health Security Agency, as it is now, but Public Health England as we know it. And essentially it just shows all these dots are levels of your antibody response to the spike protein. So this is a control group here where volunteers have been vaccinated with a meningitis vaccine. As you can see, they don't have massive levels of reaction to the spike protein. Why would they? If they've been immunized with the Oxford vaccine, this is the chimeric adenovirus Oxford COVID-19 vaccine, or the AstraZeneca vaccine as we call it, you can see that after 2 to 4 weeks after vaccination, you start to see quite high levels against the spike protein. And then if you have the prime boost, which is the second dose, you start to see even higher levels. And these levels actually are similar to levels that you see in convalescent patients, so patients that have had the disease, so almost to the level of natural infection. So my take-home message from this is have a vaccine, they work. I'm a big believer in vaccines. Okay, this is what I want to talk about work that I've been doing with the hospital over the last 10 years and many groups around Europe. So vaccines for hospital-acquired infections was the title of my talk. So this is going to be quite dry. So what is a healthcare associated infection? They can develop either as a direct result of healthcare interventions such as medical or surgical treatment, or from being in contact with a healthcare setting. Now, really, that sounds counterintuitive. You normally go into a healthcare setting to be made better and not develop an infection. But by the very nature of how some bacteria cause disease, some healthcare settings are actually high-risk areas for these pathogens. And one in particular that I'm interested in is Clostridium difficile. I'm sure you've all heard of at least Staph aureus, MRSA, but I'm going to talk about C. diff. So C. diff. All hospitals and NHS trusts have a contract. Not that I agree with this, but they do. And one of the stipulations of this contract is that they have to try and control certain diseases to below a certain threshold. And for C. diff, at the moment, the target for RD&E is 44 cases. So basically, that's what they've got to try and not have any more than 44 cases of C. diff per year. And this is in the new contracts that's been issued. But in 2016-17, if we look back, there were 31 cases of RD&E and their threshold was then 12.7. So you can see that, you know, there's a bit of well, we know this is going up. You know, so for me, I'm thinking, well, we need to be doing something about this. So C. diff, what is it? It's a gram-positive anaerobic bacterium. All that means is it has a certain cell wall morphology and it doesn't like oxygen. It's rod-shaped, and its name, Clostridium, comes from the Greek cluster, which is like a spindle. And if you look at this, this is what it does look like. It's spore-forming. This is a little endospore being formed in the bacteria here, and that is the real problem. These spores are highly resistant to disinfectants, and they hang around for ages. And also by how C. diff causes disease, which I'll come on to in a minute, that couples with its ability to form these resistant spores makes it a real problem in healthcare settings. How it actually causes disease is through the production of toxins. So tetanus is actually Clostridium tetani. So we know that a lot of the clostridial diseases cause disease through production of toxins. And for many of the clostridial diseases, the vaccines are simply inactivated toxins, as I mentioned, the tetanus toxin. So a question arises, well, you know, why is C. diff still a problem? If we know that it produces a toxin, why don't we just make a toxoid or inactivated toxin vaccine? I'll come on to that later. So if we have a look at how C. diff causes disease, it normally causes disease through disruption of microbial flora. And what we mean by that is you normally in hospital if you're poorly, and you normally get on a course of antibiotics, and those antibiotics cause disruption of your natural gut microflora, and it's a thing called dysbiosis. And during that process, it basically clears the playing field for C. diff to party and then produce toxins. And this is what happens. It colonizes, it produces these toxins, and then you have an inflammatory response, and this leads to the disease and the diarrhea. If we look at it in a bit more detail, what actually happens is the toxin enters into an enterocyte. If we look in a bit more detail, I don't want to read, you can have a look at these in the slides later if you want, but they've elucidated the molecular mechanisms of how this cell death happens. But bottom line is you end up with this horrid condition called pseudomembranous colitis or toxic megacolon. It's a really bad disease, C. diff. Okay, so as I said, there's no current vaccine. Treatment options are with vancomycin and metronidazole intravenously, not very attractive. There is the possibility of intravenous antitoxin antibodies, but this hasn't worked very well at all. What has worked very well and stops recurrent C. diff infection as well, which is a big problem with C. diff, the recurrent episodes that you have, is this thing called a fecal transplant or enema. So it sounds really unpleasant, but it's the administration of healthy poo as an enema, and that recolonizes your guts and leads to reduction in cases of C. diff. So there's a lot of work going on into understanding what's so beneficial about healthy poo at the moment, but I'm not going to talk about that. I'm going to talk about this issue that there's no current vaccine. And for about the last 20 years, there have been 3 big drug companies looking at developing vaccines for C. diff, all based around the toxins, which kind of makes sense because there's precedent for developing toxoid vaccines for clostridial diseases. Sanofi pulled out about 3 years ago now because they actually showed that their vaccine based on the toxin gave an efficacy of less than if you weren't immunized. So you're actually more at risk of developing the disease if you'd had the vaccine. So they just pulled the trial, and I thought that was quite good language. Terminated following the first planned interim analysis due to futility. It's just the other companies that are still in the race are Merck and Pfizer, and that's because they're producing antibodies against the toxins. But to me, that's not really a vaccine, because you're not going to go around administering very expensive monoclonal antibodies as a preventative therapy. And I would say the fact that these 3 big drug companies have been pumping in billions of pounds over the last 20 years and haven't really got anywhere means that we shouldn't be looking at the toxins. Okay, so my thinking was, let's have a look at this colonization area. If you can stop the bacteria colonizing, then you're just going to poo it out, so it's not going to produce its toxins, and we all live happily ever after. So the question I've always been interested in is these molecules called bacterial lipoproteins. I'll explain what they are and what they do. If you think of this as a membrane, a cell wall, this is a close-up of this cell wall, and they're these little proteins that have a stalk that anchors them into the lipid components of your cell membrane, or bacterial cell membrane in this case. Hence they're called lipoproteins. They've got lipid attached to them. They interact with the lipid bilayers. So in a way, you could think of that as the spike protein of coronavirus. And bacteria have loads of these on their surface, and are involved in all of these different processes. And the one that I'm interested in is in the middle of the slide here is adhesion. And we know that these lipoproteins, because they're expressed on the surface, might have a role in adhesion. So lipoproteins as adhesins. So we know for strep pneumoniae, there is this bacterial lipoprotein called PSAA, and that's a vaccine candidate for strep pneumoniae. ILPA of Vibrio vulnificus and BOPA of Bifidobacterium. We know that these lipoproteins are involved in adhering these bacteria to hosts. So there's also precedent of bacterial lipoproteins as vaccines. So the new vaccine for Neisseria meningitidis Men-B, that's based on a lipoprotein. There's also vaccine candidates for Borrelia, which is the causative agent of Lyme disease, based on lipoproteins. New subunit vaccines for TB are based on a lipoprotein. And this was basically the starting point for my research in C. diff. Let's go and have a look at the lipoproteins. So we can use computers to look at the genome sequence of C. diff and predict how many lipoproteins there are. And to our surprise, we went through and we found that 2% of the genome encoded for lipoproteins. In terms of numbers, that's about 70 to 80 genes that are lipoproteins. So if we want to pick one that's involved in adhesion, we didn't really have a clue. We just had a list of about 70 to 80 proteins that were lipoproteins. So this is where you go back to the literature. So we looked at this one called CD0873 because in the literature, we saw that it was recognized by patients who'd had C. diff infection. Other groups had shown that this was upregulated in vitro upon adherence to CACO2 cells. And what all that means is that when they were looking at how C. diff attached to enterocytes in the lab, they saw that the genes encoding this lipoprotein were upregulated. And also they had homology with iron binding proteins, and iron binding proteins are really important in trying to rip iron away from the host. So we were thinking, we put all this evidence together and thought, let's have a bet on CD0873. We'll go and have a look at this. So the first thing we did was we deleted the gene for this. So we made a form of C. diff that didn't have CD0873. What you can see here is these are C. diff cells. This is just staining their DNA. But if we have a mutant that doesn't express this particular bacterial lipoprotein, CD0873 on its surface, so this is our wild type strain here. And this middle panel is where we're using an antibody against this one lipoprotein, CD0873. And lo and behold, it lights up. And the fact that it lights up means that this thing must be expressed on the surface. So this is our first real lead, thinking, great, this thing in real life is expressed on the surface. And then when we knocked the gene out, it wasn't expressed on the surface. When we put the gene back in, it was expressed on the surface. So we showed that we had the tools and we could start to investigate this protein. As I said, C. diff attaches to enterocytes. It's part of its disease process. If you can stop it attaching to enterocytes, then hopefully it will just get food out and everything will be fine. So we can grow human colonocytes in the lab, and this is what they look like. And what we showed was that our wild type bacteria after an hour, so you normally get about 20 to 25 bacteria per 100 cells adhering after an hour. And then after 2 hours, you'll get, that will double to about 40. But when we deleted the gene for CD0873, what we saw was we had no adhesion, even after 2 hours. And this is better explained in pictures. So these blue dots here now are the colonocytes. The red image is the actin cytoskeleton. So this is basically what the enterocytes look like. And the green, we labeled the bacteria. What you can see with wild type, and the numbers kind of add up to what we saw in those graphs. If you just roughly think this is a 10 by 10 square, so there'd be 100 bugs, 100 cells, and then this is your number of bacteria. It's about 20 to 40 there. And then if you get rid of the gene, you see very, very few bacteria adhering to the same number of cells. And if you put that protein back in, you restore the numbers. So we were thinking this is more evidence that this protein is important in adhering this bacterium to the host. What we could also show, because we had an antibody against this surface protein, was that if this is wild type, so if we take this at 100%, this is the number of bacteria that adhere in the lab to a certain number of colonocytes. If we then redo the experiment but use antibody at quite a low dilution, we stop that adhesion. And as we dilute that antibody out, the adhesion comes back up. So that was really good for us, because we not only could we do the genetic experiments where if we get rid of the protein and stop showing adhesion, even if the protein's there, we can show that with an antibody, we can stop it sticking to those colonocytes. So this was all pretty exciting for us. We were thinking we're on the pathway to a vaccine. But then we thought, well, hold on. The reason that it hasn't worked so well looking at toxins as vaccine candidates for C. diff is because they vary so much between all the different strains of C. diff. And I think that's why those candidates haven't worked so well. So before we got too excited, we went back to many, many different species of C. diff to see first, did it have the gene for this protein? And lo and behold, it did. And is it expressed? So this is just an image where we can see that that protein, that big blob there, is expressed in all of the different types. And when we look in a bit more detail at the genetic sequence that encodes that, it's highly, highly conserved in all C. diff strains. So if you're going to make a vaccine, that's what you'd want. And we actually know now why it's conserved. I'm not going to go into that. So we published this back in, I can't remember now, this was 2014, I think, yeah, 2014. And it's, you know, it was big news. It was a major article, and it only scraped in because the reviewers said, yeah, it's all very nice. They said, but why don't you do this in animal experiments? And I thought, well, if I had an extra £500,000, I would have done that. So I thought, oh, where do we go now? And all good stories start, I think, with, I was drinking in a bar with a French lady and I met this researcher, Claire Janois, who'd seen this article, and she said, Steve, we'd love to do the animal experiments for you. So she's based in Paris, and we sent her all of our proteins and bugs, and she did this experiment where she immunized mice with the protein, and then she gave the mice antibiotics because this would then cause that dysbiosis. And then she challenged with wild type C. diff. And after certain days, she looked at the mice and looked at all their feces, looked at the numbers. But to cut a long story short, what she could show was that after challenging the mice that had been not immunized, the wild type naive mice, they lost 10% of their body weight in 2 days. Now, if you or I lost 10% of our body weight in 2 days, you know, we'd be feeling pretty sick. But if you look at the immunized mice, completely healthy. What was also really nice to see was that we could see antibodies against our protein CD873 in both the sera, but also in the feces. So it meant that we were eliciting an immune response to produce antibodies, not just systemically, but throughout the gut as well. And given that these immunizations were intraperitoneally, to get that immune response in the feces was fantastic. And we were really pleased to publish this 2 years ago now as a potential vaccine candidate. And a colleague of mine at Nottingham University, Ruth Griffin, they just published this year showing that it also works as a potential vaccine candidate in hamsters. So, you know, all I would say is watch this space and see where this goes. So that's all I want to really say. I need to acknowledge Andrea Kovacs-Simon, a wonderful postdoc in my group that did most of this work. We were funded initially by Novartis, the drug company out in Italy. And then, without a doubt, my very good friends, Claire Janois and Severine Pechin, who did all the mouse experiments and the protection studies. And I think that's about half an hour. So happy to talk and take any questions. So let's try and stop sharing.
Hi, Steve. That was fascinating to hear that and learn and hearing all about how vaccines developed in the first place was really interesting. I would just like to open up for any questions from anybody. Robin, sorry.
Hi, Steve. Thank you very, very much. Fascinating. A couple of points. I started my working career back in the late 60s with Beecham Research Laboratories. Okay, and the interesting thing was, well, a couple of things. First of all, methicillin was resistant back then. Yeah. So Beecham brought out cloxacillin and then flucloxacillin, and we're still talking about MRSA. Yeah. It's a rhetorical question, why? The thought about the French and why they're not taking, you know, the vaccines away, there's a low uptake. The French were always, I think still are, very, very keen on taking their medicines via suppositories. Now, whether that's still is, but they've got a massive suppository use in France as compared to, and then the final point was, are we as a nation confusing people because, yeah, vaccines normally produced immunity, yet the SARS-CoV-2 don't seem to produce immunity. They're almost a prophylactic. They reduce the seriousness of the condition. Because if you can still be infected after vaccination and pass on, that's not the classic definition of immunity we know from polio and MMR, is it? So I suppose that's my last question, really.
Well, it is immunity, it's just not protection.
Yeah, and that's maybe the confusion. A lot of people will think immunity is protection and vice versa.
Yeah, so that is a very good point, but I think because of the numbers and with how recently we've only known about this disease, I don't think it is just antibodies that play a role in providing protection. So I think it's kind of wrong of us just to be looking at the serological response, just the antibody response. I would imagine T cells and cytotoxic T cells play a massive role in controlling this disease, but it's too difficult really to do those experiments because you then have to be doing cell culture work in high containment labs with this virus to look at those epitopes. So I think there's a bigger story about protection that we just don't know at the moment.
A big lab in Wuhan, maybe they could be used.
Can I just ask, can you just define the difference between immunity and protection for a layperson?
So okay, I'll come back from immunity. Immunity is a bit of a loose word anyway, an immune response. So you can elicit an immune response, but that doesn't necessarily mediate protection from disease. So whilst you've still got an immune response, you might not be protected. And I think the best example to give about that is, COVID is probably the best example at the moment where, without a doubt, all these people are getting immunized. And we can see from that graph I showed, you can see high levels of antibodies, but it's not always solid protection because people still get ill, as Robin said. So, so protection is where you would do a vaccine trial and you would have an immune response which would correlate with protection. And it's really the holy grail of vaccinology. It's a correlate of protective immunity. Now that's really difficult to find, a correlate of protective immunity. And I was very, very fortunate in my 20s to go and work at the Albert Einstein College of Medicine in New York, and there were two brilliant guys. There was Bill Jacobs, who was a geneticist of tuberculosis, and Barry Bloom, who was an amazing immunologist. And they used to keep having this arm wrestle about, how are we going to get better TB vaccines? And Barry, as the immunologist, said we need to have a correlate of protective immunity. And Bill's going, rubbish, get me immunity, and then we'll go looking for it. Get me protection, and then we'll go looking for it.
That certainly seems to be where we're with COVID at the moment,
yeah,
isn't it?
Totally. Yeah.
Can I ask a question?
Yeah, please go ahead, Jill.
Yeah.
I gather that BCG is used in immunotherapy for bladder cancer, is that right?
Absolutely. So, and also I think there's a group over at RD&E that were looking at BCG into COVID as well. And the reason for that is the cell wall of mycobacteria, because BCG is an attenuated mycobacterium. And years and years ago, vaccines used to be administered with an adjuvant. An adjuvant is something that helps elicit a stronger immune response. And the classic adjuvant was something called Freund's adjuvant, but actually it was quite nasty. And they modified it a bit to something called incomplete Freund's adjuvant. And essentially all that is boiled up mycobacterial cell wall, because they have these things called mycolic acids, which are huge immunostimulatory molecules. What that means is that if you administer that, it just puts your immune system into overdrive.
Thank you.
Linda's on mute.
Yeah.
I think Linda's trying to ask a question, but she's on mute.
It reverted.
You got it.
Yeah.
Fortunately, I could hear you very well, and thank you so much for the lecture. It's really interesting. Can I just ask, is the idea that if you find a vaccine that could address what you call C. diff, Is the idea that that would just be offered to people who are going to hospital, or presumably it's not, you're not thinking of it as a kind of blanket thing for everyone?
That is a brilliant question, Linda. And yeah, that is the problem, because people don't see DIF as a problematic disease. So who's going to invest in it? And I think, you know, we have that viewpoint at our leisure and danger, you know.
I mean, the reason I was thinking of it was that, you know, most people, fortunately, don't go into hospital.
Yeah, yeah.
As we've got this huge group of people, including myself, who are very elderly, of course, as we get older, the chances are we are going to spend some time in hospital. And so I was just wondering how it was thought of in terms of the target audience, really, I suppose.
Absolutely, yeah. I mean, and I think that's why no one's really looking at it massively at the moment. I think Sanofi, Merck, and Pfizer are just, you know, they've been at it so long, they've just got to try and show some return. I think if they had their time again, sorry.
That I would much rather have a vaccine than one of the enemas you describe.
Yeah, me too.
I think a lot easier way of selling that to people, isn't it?
Yeah.
Thank you.
Especially at lunchtime, I should say, regarding the thing on enemas.
Yeah.
It's really interesting hearing about the early TB work, Steve, with the using potato slices.
Yeah,
I think
in oxbile, can you imagine? I mean, and I spent a lot of my life working on TB, and TB actually takes about 8 to 10 weeks to grow in pure culture at 37 undefined media. So how on earth they managed to keep their sterile conditions to be able to culture this? I mean, if you put a loaf of bread just in a bread bin for 5 weeks, it would be a mess.
It just shows what you can do when you need to, isn't it? Robin, sorry, I can see you have your hand up.
Thanks. Steve, you're talking about C. difficile.
Yeah.
And you said it's not Clostridium difficile, it's
Clostridioides now. Yeah,
yes,
they've got.
Please explain, sir.
It's just bacterial taxonomists. Okay.
Right. I wish I hadn't asked that. Yes.
Yeah, I know. I will always call it Clostridium.
No, okay. It's obviously one of the things which is called a nosocomial infection picked up in health environments.
Yeah.
Are there any other nosocomial infections that need vaccines or need some work on to reduce?
Definitely. So the big problem ones at the moment, so those figures I took about thresholds from the contracts with NHS, the other bloodborne infections that they worry about are there's two in particular, gram-negative bacteria called Klebsiella and Acinetobacter. They're becoming a massive problem.
And are there antibiotic treatments for them, or is it still?
Well, funny you should say that. There are, but they're resistant to a lot of them. But we've got a project here, and I will shamelessly send a link around. So we've got a project called Citizen Phage Library, and what we're trying to do is look for bacteriophage in the environment. So bacteriophage are viruses that will kill bacteria, and they're ubiquitous on Earth. They're everywhere. So we just got to find them. So what we do is we try to engage the public by sending them out a little pot. They can go to a water course, send it in, and then we'll extract the phages, purify them. We'll do electron microscopy. We'll give the sender in, we'll give them a name. So our best ones at the moment, we've got one came from a chicken house water feeder, and we've called it Kylie Mineg. And we've got another one called Lemonade. But yeah, I'll send that link to Kim and Louise, and they can send it around to you all. And feel free to share it.
Yeah. Because I mean, if you're looking for, there is this extra. I'm part of how I've come to this group. There's the Exeter 10,000, the research group. Have you approached them to go out with sampling devices?
No, but I'm a massive fan of the Exeter 10,000 group, and I think we should give a little nod to Jill Baker here.
Yeah, because there are those people we're all signed up to believe in research, I guess. So
yeah. Well, I mean, if you're involved with the Exeter 10,000, yeah, that's a good point. I hadn't thought about that. But I will send you the link to the Citizen Phage, and please do disseminate it.
Thank you.
Yes, certainly disseminate it to the PPI groups, and perhaps Louisa, we can perhaps see about whether we can put it in the next COF newsletter or something. So I'll have a chat with my colleagues about that, because if we get it in the newsletter, it might just open it up to more, which goes out to the extra 10,000 participants. That might open it up.
That'd be great.
A bit more people. Yeah, I'll see what I can do about that, and I'll be in touch with you about it.
Jill Haynes, no, it's a good question. No.
Okay. Is there any other questions for Steve at the moment? Okay. I would just like to thank you so much, Steve. That was a really fascinating talk, and really,
it was really fascinating.
Absolutely.
Thank you so much for your time.
Brilliant.
Really good.
Thank you.
Really useful, and it is just and as I said, fascinating hearing how it's all kind of developed over the ages as well. And also fascinating to hear you used to work at Porton Down. I did my placement year for my degree at Porton Down, so yeah, got great memories of my time there.
Was that a Public Health England or MOD?
MOD.
Yeah.
My son's working there now.
Oh, is he?
Well, actually, he's working upstairs, but he's at Porton Down.
Small world.
It's bizarre. There's two institutes there. Well, Health Security Agency now, and DSTL as it is.
Yeah, he's DSTL.
But
yeah,
they don't talk at all. It's like they could be different parts of the country.
I know, and they're just literally next door, isn't it? They're so close.
Yeah. It's the same road up off the plains. Yeah.
All to do with the health security bit. You know, the security word they've now got in.
Yeah.
Who knows? Who knows?
Anyway, nice to see you all.
Thank you.
Thank you very much.
Also, people just to stay on the line in a moment, just to give me any suggestions for the next slot of training topics that you would like to have. That would be really useful. And I will email Steve later and just reiterate our thanks to him. I just wanted